Hello and welcome. My name is Liliya Gilroux, and this is my presentation on cave-dwelling arthropods of Rapa Nui, Chile. Rapa Nui, also known as Easter Island, is a Polynesian island over 3,500 kilometers west off the coast of South America. Due to its size, topography, geographic isolation, aridity, and anthropogenic-driven activities, this island has undergone extensive environmental change. These changes culminated in a catastrophic ecological shift, transforming the island from once a palm-dominated scrub forest to now largely a grassland community. Additionally, during the mid-19th century, an island-wide sheep herding operation exerted additional pressure, drastically altering the island's fragile ecosystems. Due to this complex and principally detrimental environmental history, most of the island's terrestrial endemic fauna has gone extinct. Today, remnants of the island's endemic terrestrial fauna are likely to be only invertebrates. Caves were intensively examined because they represent relict habitats and are believed to be somewhat buffered from the extensive environmental changes that occurred on the surface. Research over the past 10 years revealed that Rapa Nui caves harbored endemic species. To date, 10 endemic cave-dwelling arthropod species have been confirmed. 8 of the most recent discovered endemic species are featured in this photo. We anticipate this research will ultimately result in the discovery of additional new endemic species. 18 caves were studied between the months of July and September of 2016. Field researchers applied a range of collecting techniques, including bait stations, baits including fish, branches, and sweet potatoes, leaf litter traps, time searches, direct intuitive searches, and opportunistic searches. Sampling was limited to the estimated cave deep zones and the fern moss gardens occurring in the cave entrances. A cave deep zone is characterized as completely dark, near constant ambient temperature, little to no airflow, and a water-saturated atmosphere. From an evolutionary perspective, deep zones are tremendously important, as it is the only environmental zone where subterranean adapted animals can evolve and exist. In addition, fern moss gardens within cave entrances have been confirmed as relict habitats supporting 10 endemic, tentatively cave-restricted arthropod species. With over 1,000 specimens collected, our preliminary findings show at least 47 morphospecies were identified across 18 taxonomic groups. For those of you wondering, what is a morphospecies? A morphospecies is a taxonomic grouping of individuals based on morphological or physical similarities. So far, at least 3 of our cave-dwelling morphospecies may represent both species new to science and potentially endemic species. More specifically, our findings show that spiders are the most speciose group, with at least 14 morphospecies identified. The second most diverse group are beetles, with 6 morphospecies. The remainder of the 16 taxonomic groups have 3 or fewer morphospecies per group. The number of morphospecies in our collection will likely fluctuate until all specimens are identified to the lowest taxonomic level possible. Hopefully this means we will be able to identify most of the morphospecies to the species level. Additionally, it is important to note that the 10 endemic species previously believed to be restricted to caves have not been confirmed from any of the 18 caves examined during this 2016 survey. As this work continues, we will further examine and identify all species to the lowest taxonomic level. We will also identify morphospecies believed to represent new species and send these specimens to taxonomic specialists for confirmation and ultimate description. Once identifications are complete, we will then calculate species richness estimators, compare diversity across caves, potentially expand the range of known endemic species, and likely identify additional endemic species new to science. Ultimately, these analyses will improve our understanding of endemic arthropods of Rapa Nui. Conservation and management practices of caves should be continued and prioritized. Once the study is complete, these results may be used to further educate the Rapa Nui community regarding the importance of endemic cave-dwelling species, as well as heighten public awareness concerning the impacts of unregulated adventure tourism. In addition, as caves are highly challenging environments to effectively sample, additional sampling at these sites are recommended. Finally, endemic cave-dwelling arthropods represent the last remnants of Rapa Nui's native ecosystems. Therefore, these relict habitats and their associated arthropod communities should be periodically monitored to both detect changes to these habitats and to potentially detect and manage alien arthropod species that could challenge the persistence of these relict populations. Finally, I'd like to acknowledge the NAU Office of Undergraduate Research and Creative Activity via the Interns to Scholars program for funding the development of this research. Thank you.